heard that the human genome has tens of thousands of genes in it, but did you know that not all of these genes are expressed? Cells carefully and strictly regulate when and which genes are turned on and which ones are turned off. This is what we like to call regulation of gene expression. There are many different kinds of gene regulation, but today we are going to focus on just one, gene regulation at the level of transcription. First, we are going to talk about gene regulation in bacteria. Bacterial genes are often found in operons. An operon is just a fancy way of saying a cluster of genes that regulate other genes. Every operon has a promoter. The promoter is where RNA polymerase binds and begins transcription. Once the promoter is activated, an RNA polymerase binds to start transcriptions. Genes 1, 2, and 3 are all transcribed into mRNA. This is because in this operon, the promoter regulates all 3 genes. Next, we will go over the anatomy of the operon. Operons aren't just made up of the coding sequences of genes. They also have regulatory DNA sequences that control transcription of the operon. These sequences of DNA are binding sites for regulatory proteins. Regulatory proteins control how much or how little the operon is transcribed. The promoter is an example of a regulatory DNA sequence because it can start or stop transcription. Most operons have other regulatory DNA sequences in addition to the promoter. These sequences are binding sites for regulatory proteins that can turn expression of the operon up or down. So there are 2 types of regulatory proteins that bind to these regulatory DNA sequences. The first are called repressors. Repressors bind to pieces of DNA called operators. When bound to its operator, a repressor reduces transcription, meaning it stops or slows down the transcription of the genes. It does this by blocking RNA polymerase from moving forward on the DNA. The second regulatory proteins are called activators. When an activator is bound to its DNA binding site, it increases transcription of the operon, meaning it starts or speeds up transcription of the genes. It does this by helping RNA polymerase bind to the promoter. Many regulatory proteins can themselves be turned on or off by specific small molecules. The small molecule binds to the protein, changing its shape and altering its ability to bind to DNA. For example, an activator may only become active, meaning it's able to bind to DNA, when it's attached to a certain small molecule, as seen here. Operons can be either inducible or repressible. This is just a fancy way of saying that some operons are usually on, while some operons are usually off. Some operons are usually off, but can be turned on by a small molecule. The molecule is called an inducer, and the operon is said to be inducible. An example of an inducible operon is the lac operon. The genes in the operon encode proteins that allow the bacteria to use lactose as an energy source. To use lactose, the bacteria must express the lac operon genes, which encode key enzymes for lactose breakdown. When lactose is not present, no transcription of the lac operon occurs, and this is because the lac repressor remains bound to the operator and prevents transcription by RNA polymerase. When lactose is present in the cell, strong transcription of the lac operon occurs. The lac repressor is released from the operator because the inducer is present. Other operons are usually on but can be turned off by a small molecule. The molecule is called a co-repressor and the operon is said to be repressible. An example of a repressible operon is the trip operon. Tryptophan is an amino acid that bacteria, like E. coli, use to build proteins. Bacteria can get tryptophan from the environment, but if it is not available, then it will make its own. The genes that encode for tryptophan are located in the trip operon. When tryptophan is present in the cell, it attaches itself to the repressor molecules, which change their shape and activate them so that they bind to the DNA, physically blocking RNA polymerase from moving on and transcribing the genes that make tryptophan. This is because there is already enough tryptophan in the cell, and it would be inefficient for the cell to make more. When tryptophan is low or not present in the cell, the trip repressor is inactive because no tryptophan is available to bind to it and activate it. Also, the repressor does not attach to the DNA because it's inactive, so RNA polymerase transcribes the genes and makes mRNA that will code for tryptophan.